So we've got, I think, about 5-6 minutes to take a few questions from the audience for the presenters, and I think John is intended to stay on the line, so hopefully he's still there. Uh-oh, we got trouble from the get-go here.
Jim Stevens, microphone is not on.
That was by first intent, just so you know. John is on.
Microphone is on. Mundane question. If you're in pharma, you could spend 5 days a week having companies come through and pitch you their latest microphysiological system, or academics. And fortunately, we've developed a couple collaborations with some well-known and very bright microphysiological system scientists, some of who are in the room and on the phone. But what can the community do to actually establish standards? In other words, if we say, I've got a good liver system, I got a good lung system, I got a good heart system, is there a possibility that community can actually define what good is in a quantifiable way so that when a company comes in, we can actually compare apples and oranges to try to make a decision? This is the one that, to us, replicates physiology we care about, whether it's from an efficacy point of view or a safety point of view. I think there's something out there, but could you guys address this idea of standards and what defines good?
I'll start, although I know John will have something to say to this. I'd start by saying that we're still pretty early in the evolution of human microphysiology systems. So I think that we're going to see, especially over the next 5 years, continued evolution in terms of reproducibility of the different organ systems and the kinds of things that they can do. So I think this is the time where we should start talking about standards. The first step is what the NCATS is doing through the tissue chip testing centers, is just to demonstrate initially reproducibility in any of these chips. In time, I think the commercial drivers will kick in order to define a system, and the public will then have to evaluate those.
May I add something?
Yes, sir.
Okay, I agree totally with Lan. The paper you need, or the presentation you need to listen to in answering that question is going to be Jim Stevens later in the morning, because they did a study that showed that a mouse liver was a better model of a rat liver than rat hepatocytes in a dish, and that rat hepatocytes in a dish looked like a rat that had been exposed to a high level of toxin. The bottom line on your question, which is totally fair, is you have to decide what question you are answering and how good a model do you need to answer that question. A mouse is a perfect model of a mouse, and a microphysiological system is a toy model of a human. It'll never be as good as a human. It'll probably be better than a well-plate or a petri dish. But you really have to decide what you are trying to measure. And again, I think the stuff that Jim is doing is what needs to be done, in addition to the tissue chips testing centers.
Well, thank you, John.
Do you guys prearrange that?
I was going to say, were you fishing for that, Jim?
John, that was actually Jim who asked that question.
Oh, that was Jim that asked the question. Hello, Jim.
Rob Burgess from Jackson Labs. I have a related but maybe kind of fundamentally earlier question that maybe should be addressed to Dr. Burridge or McCain. Related in the iPS cells themselves, what's the impact on reproducibility of whether the cells come from blood versus skin, whether your reprogramming protocols, your differentiation protocols, and is there effort in the field to try to standardize those methods?
So I can, whether it's blood or fibroblasts, we get this reviewer's question all the time, and it is absolutely zero. There is no difference whatsoever. The two studies I described today, one was done from fibroblasts, one was done from blood cells. They're iPS cells. They're exactly the same. There was a theory in the field for a while about this idea of epigenetic memory, and I think that has been thoroughly debunked. There is, but only in very, very early passage cells. The cells we work with, nothing at all. When it comes to the differentiation, it's fascinating in the cardiac field that many of us use a variety of different differentiation techniques, but when we look at the transcriptome or the electrophysiology of these cells, they all come out the same, which is not what we would expect. But yeah, today it seems the cells are all largely pretty much the same. I think that's going to change as we improve maturation and subtype specification, but for now, yeah, very similar.
Yeah, let's see, I'm on? Yeah. So just to add, I think one of the biggest challenges for us is just patient-to-patient variability, but that's kind of hard to overcome, and at least with CRISPR-Cas9 and the isogenic controls and things like that. So I think it is critical for every group to do thorough kind of characterization and purification, and a lot of the protocols developed by Paul and others in terms of making these higher pure systems, and a lot of work in maturing these cells and getting them to a more adult-like state will also hopefully improve the state.
So this will be our final question before break.
Joe Newsom, University of Pittsburgh. Wanted to sort of expand on Jim's question. Part of this whole attempt we're doing today and yesterday was to evaluate where we're going with precision medicine and as it relates to animal modeling. And I was wondering if the panel could speak a little bit about sort of like what Lance, you said. I learned quite a bit from a GLP tox study using an animal model, and I learned a lot from my in vitro models as well. Where are the challenges to make a decision about what's going to be best, or are we going to continue to need both? So I'll just open that up for the panel to comment on, please.
Could you repeat the question, Brian?
Yep. Yeah, so the question was that we recognize that there are lots of things that we learn in our traditional animal studies, and then they're going to remain a necessary part of our business. But we're also recognizing that there are some distinct things that we can learn in these microphysiological systems. And so ultimately, how do we integrate them? Is that a fair representation? Integrate them, and so that they're complementary and part of the evolution in potentially how we do the animal study?
Or is there a timeline that people are anticipating? I know we have a lot of validation to do with these systems, but is that 10 years from now?
Right.
50 years from now?
And a timeline.
What's the timeline?
Does that make sense, John? Did you?
Yeah. I mean, the problem that is faced with microphysiological systems is the same problem that's been faced for decades on in vitro to in vivo extrapolation. So you have to extrapolate from cells grown in a dish to a human dosing, and there's going to be the same problem that pharma has addressed over the years. Those techniques have to be put together, and we'll probably have them. I think Lance will probably have it finished tomorrow.
Sure.
So I think one valuable fact is that the FDA has joined with the EPA and the NIH and NCATS in taking part in the microphysiology systems program. And they are particularly interested in seeing how far the MPS systems can advance. And the projection is, not promised on their part, but the projection is at some point where the devices are deemed to be reproducible and have enough function to be really useful, the FDA is probably going to encourage co-animal studies and in vitro studies. And I think that's probably over this next 5 to 10 years is my projection.
So one of the ideas that, so I'm involved with a pharma consortium group partnered with, in fact, the folks in NCATS and Lance and John, and in sort of looking at that. And one of the things that we've socialized is this notion of the evolution and our confidence in these systems and understand what they do and don't tell us. And one of the beauties of the safety system that we have, and I'll talk a little bit about this in the next presentation, is that there's some redundancy built in there. I mean, you could envision a day where we've got single animal species coupled with microphysiological systems for which we've gained some confidence in their ability to tell us meaningful things that are part of that evolution. I don't know what the timeline is, but I think that could be a part of it. Megan or Paul, anything to add to that?
I see them as working side by side for quite a while. So there's questions and answers that you can get from each model system that I think you can't get from the other. So in terms of immune response, right, and kind of off-target effects of drugs is going to be really difficult to model with microphysiological systems in the near future. So I think there's really a lot of unique things to learn from both systems, and doing both in parallel is how I see the foreseeable future, at least.
With that, I think we'll take our break, and we're due back from break at 10:30. So thank you for your attention. Thanks, John.
Thanks, Brian.